Glycosidic bonds are the bonds that exist between the individual monosaccharides in any given polysaccharide. And of course, we can also have glycosidic bonds between sugars and proteins or between sugar and lipids. Now, anytime we have a glycosidic bond formed inside our body, that reaction is catalyzed by a specific type of enzyme. And the enzymes that catalyze the formation of glycosidic bonds are known as glycosyltransferases. Now, let's take a look at the reaction that is actually catalyzed by this type of enzyme. So what exactly does the reaction actually look like? So let's suppose we have a sugar, and the sugar is basically shown in red. And what we want to do is we want to attach another sugar, let's say a glucose molecule, onto that red sugar. So this is the purple glucose that we want to attach onto that sugar molecule. Now, before the glycosyltransferase can actually catalyze this reaction, what we have to do is we have to activate this incoming glucose molecule. So we have to activate it, we have to make it more reactive. And the way that we make it more reactive is we add a nucleotide component onto this glucose. Now, how exactly does that make it more reactive? Well, by adding the nucleotide, we add these negative charges. And anytime we add negative charges onto a system, we make that system more reactive. Its energy increases, and that makes it more reactive. And so we basically activate the sugar by adding that nucleotide component, and now we have an activated glucose nucleotide, and now we can use the glycosyltransferase to basically catalyze the formation of the glycosidic bond between this carbon of the incoming glucose and a carbon on that sugar molecule. Now, in this particular case, because this is glucose, this molecule is known as uridine diphosphate glucose. Now, once we form that bond, this is the bond that is formed shown in green. That's the glycosidic bond. And of course, instead of having the sugar here, we can also have a protein or we can have a lipid. In either case, we form a glycosidic bond as a result of the action of glycosyltransferases. So glycosyltransferases use an activated sugar, usually a sugar nucleotide, such as in this case, uridine diphosphate glucose, to basically catalyze the formation of that glycosidic bond. Now, just like any other group of enzymes, glycosyltransferases are highly specific molecules. They basically catalyze specific sugar reactions. They use specific sugars to form glycosidic bonds. So what exactly does that mean? Well, to demonstrate what that means, let's take a look at the ABO blood type that exists inside our body. So let's remember a bit of biology. So any individual either has blood type O, blood type A, blood type B, or blood type AB. So what exactly differentiates one blood type from another blood type? So essentially, on the membranes of our red blood cells, there are specific antigens, more specifically specific glycoproteins that are expressed. And it's the type of glycoprotein on the cell membrane of the red blood cell that determines your blood type group. So let's take a look at the following three diagrams. So this is the cell membrane of our red blood cells. Now, in an individual who has the O type, they have the O antigen. In an individual who has the A type blood, they have the A antigen. In the individual who has the B type, they have the B antigen. And in the individual who has the AB, they have both A and B antigens. Now, this is the protein component of the glycoprotein, the antigen. And notice the protein component is exactly the same in each one of these cases. In fact, we also have these carbohydrates attached onto the protein component, and that's what makes them glycoproteins. And notice that all these three antigens have a common sequence in common. So this sequence is also found here as well as here. So what exactly is this sequence? Well, these blue triangles are galactose sugars. This circle here is the N-acetyl glucosamine, and this square here is the fructose. So we have this oligosaccharide sequence known as the O-oligosaccharide sequence that exists on the O antigen, and we also find that on the A antigen and the B antigen. So what exactly is the difference between these three antigens? Well, it's basically the absence or presence of an additional sugar molecule. In the case of the O antigen, this is all we have. In the case of the A antigen, we have an additional sugar that is linked via the alpha-1,3 linkage between the galactose and this N-acetyl galactosamine. In the case of the B antigen, we have this galactose linked via the alpha-1,3 linkage to another galactose, and that's what differentiates these three types of antigens. So we see that the antigens in the ABO system are O-glycosylated glycoproteins, and what that means is not only do we have the protein on the antigen, we also have this oligosaccharide as shown in this diagram. Now, it turns out that the terminal sugar residues on the antigen determines the type of blood group, as we discussed just a moment ago. So if we have this type of linkage and this type of additional sugar, that's the A. If we have this one, that's the B. If we have neither, then it's the O. So we see that all three antigens have the O-oligosaccharide sequence in common. Now, what ultimately determines if an individual has one of these three types of antigens, or if they have both of these antigens? Well, basically, it's the genetic information in the DNA. If the individual contains a gene that codes for a specific glycosyltransferase that is able to actually form this bond, then they're going to have the A antigen. If they have that specific gene that expresses the glycosyltransferase that creates this bond, then they're going to have the B antigen. If they have both genes that basically create both of these glycosyltransferases that form both of these linkages, then they're going to have both of these antigens, and so they're going to have the blood group AB. On the other hand, if the gene is absent from that individual and the individual cannot synthesize either one of these glycosyltransferases that can actually form these two types of linkages, then they're not going to be able to form the linkages, and so they're going to have this molecule here, this glycoprotein. In that case, they're the O blood type. So we see that a specific glycosyltransferase must be present in order to add an extra sugar to the galactose of the O-oligosaccharide sequence, this 4 sugar sequence shown here. So a type A glycosyltransferase is needed to add the N-acetyl galactose, while a type B glycosyltransferase is needed to add the galactose to that O-oligosaccharide. And blood type O individuals basically have genes that cannot express either one of these, type A or type B glycosyltransferases, in which case they cannot form these bonds, and so they end up having this glycoprotein on the red blood cell membrane. So we see that glycosyltransferases are highly specific molecules, and inside our body we have these specific glycosyltransferases that can catalyze the formation of specific bonds between specific types of sugar molecules.